Hello, good day, and welcome to you all to this webinar, and welcome to you, Derek.
Thank you, Ivan.
So this is a series of webinars hosted by Alphalize on the analytics for CMC with special focus on HCP analysis by Mass Spec. So my name is Ivan Mertz. I'm the Chief Operating Officer of Alphalize, and my passion is mass spectrometry for host cell proteins. So I'm very excited to have you all for this webinar today. I'm your host today and the moderator. The talk by Derek will take about 30 minutes, and then we will follow by a live Q&A where you can ask Derek questions about his talk. So if you have questions during his presentation, then please type them in the chat, and we will go through them afterwards. The topic of today is the new USP chapter 1132.1, using mass spectrometry for host cell protein analysis, and the standards to be provided by the USP to support these analysis. This is a very exciting topic for many people. We have more than 400 sign-ups for this webinar today. So we think, Derek, that this is going to change the industry as we think about it today. So just a short presentation of Derek. Derek is a senior scientist from the US Pharmacopoeia, where he's leading the development of reference standards and analytical reference materials for biologics. In previous roles at Adaptive Phage Therapeutics, Derek oversaw the analytical development and qualification of potency and characterization assays, including LC-MS and ELISA for HCP analysis for drug substance release. And at United Therapeutic Corporation, he managed assay development for drug substance characterization, including validation of process-specific HCP ELISA for GMP release testing. So clearly, Derek, you are an expert on this topic, so we are looking forward to your talk today.
All right. Well, thank you so much. Well, thank you, Ivan, for the kind introduction. My name is Derek. I'm from US Pharmacopoeia. I'm with US Biologics. And without further ado, I will start my presentation. All right, thank you and welcome to my presentation. And today I'm going to be giving a talk on USP's initiatives for enhancing the quality and consistency of mass spec-based host cell protein analysis. So just some brief outline of what this talk is going to be about. We're first going to look into the significance of host cell protein. And then we're going to do a little brief introduction on what are the USP standards and what are USP standards for host cell protein. And then we're going to dive deep into the chapter outline and highlights for the new 1132.1 chapter on LC-MS for host cell protein analysis. And then we're going to look at some of the host cell proteins physical standards that USP is currently developing. And lastly, there will be a summary slide at the end. So just a brief introduction to US Pharmacopoeia and Compendia for those who are not familiar with US Pharmacopoeia. So for over 200 years, US Pharmacopoeia has provided public standards to support the quality of medicine and improve public health. USP, we are an independent scientific nonprofit organization. We're focused on building public trust in the supply of safe quality medicine. USP, we have over our standards are used in over 150 countries and enforced as requirement by law in over 40 countries, such as our USP NF dietary supplements and also the FCC. And our global efforts is to ensure testing and public standards that are made available to verify quality and safety of biologics. And USP Biologics, we are working on expanding our standard developments and testing to support all life cycles of biopharmaceutical products. Now, let's talk more about host cell protein. So why do we care about host cell protein? Well, obviously, if you're in this webinar, you already probably do a lot of characterization and measurements with host cell protein, or you're doing bioprocessing, trying to get rid of certain problematic host cell protein. So recombinant proteins expressed in host cell lines, such as monoclonal antibodies, insulin, and other biotherapeutics, and these mostly are commonly expressed in systems, cell line systems such as CHO hamster ovary cells, E. coli, and mouse cell lines. And for the more novel therapy that's coming into the market nowadays, they're expressed in HEC, SF9, Vero, and more novel cell lines. And residual host cell protein have potential to affect both product quality, safety, and efficacy. Host cell protein can elicit unwanted immune responses. They can also have bioactivity, which are homologous to endogenous human protein. Host cell protein can also be enzymatic, which can cleave drug substance or excipients that affect stability and potency, such as some of the enzymes that are commonly seen are lipases and proteases. And these risks are varied based on many factors, including the dosing, route of administration, the frequency of dosing, whether or not the patient is receiving more than one biotherapies, and patient population, and different patient will have different immune responses. And then what are some of the strategies for monitoring host cell protein, right? So the most common one that we see is the commercial ELISA kit. These are commercial ELISA kits with limited antibody coverage, but they're pretty good for initial development during the early clinical phases. And the measurement output is total HCP in nanograms per mg, which is relative to the product. And then during a later phase development, a lot of biomanufacturer will go into more process-specific ELISA. These are immunogens from manufacturers own no host cell HCCF or lysate. And the final measurement output is total HCP in nanograms per mg, which are quantitative relative to the target products. And orthogonal to ELISA, there is the mass spec. So this can be described into two different type of mass spectrometry. One is the untargeted, and others being the targeted or target-specific mass spec. With untargeted, you can have both identification and relative quantitation of individual host cell protein as well as total HCP. These are wide in dynamic range, but also requires a lot of technical skills to set up. And with target-specific mass spec, this enables both identification and precise quantitation of a single target HCPs. And these are requires quantitative calibrants, such as heavy cell-labeled peptides. So what are USP standards for those who are now familiar with our standards? So USP standards is divided into two types. One, we have documentary chapters, which are documentary standards. These are divided into procedure chapters below 1,000 and more informational chapters if they're above 1,000. So for the examples of more informational chapters are such as 1132, which is the residual host cell protein measuring in biopharmaceuticals for ELISA, which was published back in 2016. And we also have the 1132.1, which is the upcoming one for residual host cell protein measurement in biopharmaceutical by Mass Spec, which is expected to become official May 1st, 2025. And then we also have physical standard along with documentary standards. These are divided into reference standards. Reference standards ensures identity, strength, quality, and purity of pharmaceutical articles and can be linked directly to a procedure in the US Pharma Compendia. Then we have analytical reference material. These are relatively newer. They're not compendial, but they're still well-characterized materials and can be used for fit for purposes. Now let's dive into the chapter outline and highlights from 1132.1. So why was this chapter necessary? So it's for the past 5 years, the mass spec analysis of host cell protein has really matured. And as we stated originally in the USP chapter 1132, that host cell protein analysis by immunoassays, specifically ELISA, is very reagent specific and cannot be standardized across the industry. Well, ELISA is still the most used methods. Results are linked to proprietary reagents and report only the total HCP. With the most recent advances in mass spectrometry methods and widely available whole cell protein materials, this has really changed the testing landscape, which enables comparison results. And this is specifically true, which enables some simultaneous identification and quantitation, which makes mass spec a powerful orthogonal approach. This new chapter is really aimed at providing both new and experienced user informations and best practices for developing or refining their LC-MS, MS-based approaches for product development. It is intended to expand and improve the whole cell protein toolbox along with their ELISA. So this is just a highlight of our whole cell protein expert panel. This expert panel was formed back in beginning of 2020, right in the beginning of COVID-19. And our expert panel consists both expert volunteers, government liaison, as well as USP scientific liaisons. From expert volunteers, we have, I'll just name a couple few of the people here, like Ned Severin Clavier, and then Kevin Van Cott. We also have Feng Wang from Merck, and also Yang from Sarepta. These are very well published authors, and some of their papers you probably still are using in your own lab as references to develop your methods currently. So we selected our expert panel really based on their high credentials to write this chapter. And then this is just a brief chapter outline. I'm not going to go through every one of these, but I'll highlight a couple of them, which we're going to dive deeper into. So some of the important parts from this chapter is really the sample preparation, chromatographic separation, mass spectrometry analysis that's described in the chapter. And then also the quantitation approaches that's outlined in the chapters. And then also the best practices for reporting ELISA versus mass spec data, which we'll talk about in this presentation. And you can find the link to the PF in the link below. So first, let's talk about sample preparation as described in the chapter. So this involves, basic protein samples using mostly trypsin to be digested in various conditions. And in the chapter, we described 3 different types of sample prep, one being product depletion, second being standard digest, and last being whole cell protein HCP enrichment. Product protein depletion usually, as described in the chapter, we talk about it as native digest. This also involves absorptions onto affinity columns, and you can also do 1D or 2D SDS page gel analysis or LC fractionation of proteins. And standard digest is your standard denaturation, usually with reduction alkylation and digestion of raw proteins in the sample. And the whole cell protein enrichment being the third is also another strategy that can be used for sample prep, where polyclonal antibody against the whole cell protein is bound to a column such as AAE, and then affinity reagent and enzymatic activities are used to further enrich the whole cell protein. And then the second part of the chapter that we really dived into for this presentation is the mass spec workflow. So the typical LC-RM, MRM workflow, which I'm going to describe here is, and we'll talk about later in this presentation, is first we have the LC separation, followed by electrospray ionization, and then typically this is followed by MS1, either DDA or DIA, and then followed by CID fragmentation, which goes into MS2, and then detection. And last part, but not least, is the data analysis portion, where we described in thorough details in the chapter, which we believe to be very important. We identify 3 different quantitation strategy, one being relative to product protein, second being relative to spiked in proteins, and third relative to spiked in peptides. So with respect to product protein, a known amount of products is used to quantitate the amount of whole cell protein by compared signals. The relative to spiked protein strategy uses intact protein with known concentration used as standard to quantify the whole cell protein. And relative to spiked peptides, are peptides with known concentrations and are spiked into samples and used as calibration standards. So this is probably the most interesting part and probably one of the most important parts about this presentation is reporting results. So how should whole cell protein content be reported and what units are appropriate when you're using mass spec? So, well, first, the easiest way is to list identified protein, simplest output, but not quantitative, right? And then you can also list of identified proteins with concentration. This requires conversion of molar results to mass using whole cell protein molecular weight and express results as nanogram HCP per mg of product. Second thing that we thought would be important is also to include the sensitivity of method capability. Average sensitivity can be established through spiking studies using intact proteins. A variety of approaches can be used for qualification of sensitivities. And the last part is ELISA and mass spec comparison. These are very complementary methods, but the correlation of LC-MS and ELISA for a single protein can be established if standards are available. So this is a very important part, last part here, is that when you're assessing your ELISA and mass spec, you need to take very careful approach in drawing conclusion or attempting to match the numerical results for total HCP. As ELISA HCP and LC-MS quantitation principles are very, very different, even though they are complementary. Now I'm going to talk about the physical standards that USP has been developing to support identification and quantitation of high-risk HCPs. So from our stakeholders, we have identified there's a need of standard to support greater consistency of quantitation across labs. We are focused on individual proteins or peptides corresponding to HCP of concerns. These HCPs have been reported to impact patients or product or are persistent in purification and has been verified against published benchmarking studies. And we are dedicated to support identification and quantitation with mass spectrometry being our first priority. So for individual whole cell protein, we have released our CHO PLBL2, which has been made available since last year. And we're currently in the workflow of our CHO no cell HCCF, which is currently in development. And we also have the cell peptides for individual whole cell protein, which we will dive deeper into in this presentation. So individual whole cell proteins of concern. So PLBL2 is a pretty notorious high-risk HCP. So we have a recombinant, at USP we have a recombinant CHO phospholipid B-like-2 protein. And this is a C-terminal 6-his tag expressed in CHO protein. And we have made this in 2 mg per mil formulated with 1x PBS with 100 microliter per vial. And this is a very well characterized both by appearance, concentration, purity, identification. And some of the additional testing that we did not include in the product information sheet include peptide sequencing and mass spec. So on the right-hand side, you can see the purity analysis by SEC as well as the identity by SDS page. So some of the potential application for this whole cell protein of concern is for usage in ELISA, Western blotting, control for development of MRM for PLBL2 and identification and quantitation, and also can be used as a LC-MS system suitability testing material. Now let's dive deeper into the cell peptides for individual whole cell protein. So this is a general diagram that for whole cell protein testing strategy if you're using cell label peptides. So this workflow really starts with protein digestion, typically by trypsin, followed by the separation of peptides using liquid chromatography and analysis of separated peptides by tandem MS. And then USP's reference peptides can be used to aid in identification and quantification of their corresponding host cell protein. Specifically, when using relative to peptide quantitation approach, as described in the chapter 1132.1, because of the distinct isotopic nature of these peptides, they can be added directly into samples without interfering with quantitation of non-labeled protein from the digested host cell proteins. So the USP, so we have been working on these cell label peptides for the past 2 years. These peptides are very well characterized, and some of the specifications that we have for these peptides are they're 1 mg per ml dissolved in liquid, and they're stored in -65. And some of the characterization that we have done are peptide content by AAA analysis, purity by HPLC UV greater, and we aim for greater than 95% purity, and sequence identity by LC-MS. These peptides are aimed for application for mass spec workflow for both identification and quantitation. We have already released, and these products are already available for both clusterin and lipolipase, and there are 3 peptides each for these 2 host cell protein of concern. And next year, we'll have the second round of release, which 6 additional host cell proteins of concern with 3 peptides each. And then we also developed our own internal case study using on USP peptides. Right? This is a developed LC-MRM method to quantify both the LPL and clusterin using USP cell label peptides. So the sample preparation that we took is a pretty standard denaturation, alkylation, and trypsin digest. And here below you can find the LC parameters. We use the waters acuity, UPLC, HSS, T3 columns, and then the column temperatures at 40 C, flow rate 0.2 ml per minute, and the mobile phase A, 0.1 formic acid in water, mobile phase B being 0.1 formic acid in acetonitrile with injection volume of 20 microliters, and autosampler temperature of 4 C. And then this is followed by a triple quad mass spectrometer using electron spray ionization in positive mode, and quantitation is using relative to spiked-in cell-labeled peptide approach, as we described in the chapter 1132.1. So for each of the cell-labeled peptides and associated unlabeled peptide, an MRM-MS method was developed. So you can see the MRM parameter in the table above, which shows the precursor ion, the parent ion, and the daughter ion, as well as the rotation time. And you can find the sample chromatogram for each of the cell peptides in the chromatogram as displayed in this slide. And then we also assess the feasibility of this internal LC-MRM method that we set up using USP cell label peptides. So the first thing that we assessed was the specificity of the cell label peptides. So we analyzed the following sample using the cell label peptides in diluent, 1 nanomolar cell peptides in diluent, and samples including USP map 003, CHO HCCF, CHO protein A pool, and CHO IEX without spiked-in cell label peptides. So in specificity, sample lacking the spiked-in cell label peptides, no cell label peptides peaks were observed at the expected retention time using our MRM method. So, and then we assessed instrument precision, which is the repeatability. This was investigated by preparing a sample containing all 6 cell label peptides at concentration of 1 nanomole per peptide, and 6 injections of the sample were tested and analyzed on LC-MS. And all percent RSD falls within less than 5%. And then we also assessed the method precision. This was evaluated by calculating the RSD of the peak area of each of the cell label peptide from duplicate injection with 3 preparations. And each preparation contained 1 nanomole of individual cell label peptides in a USP map solution. And we can see the percent RSD is around less than or equal to 10% for method precision. And then lastly, we also assess the LLOQ of each of the cell label peptides. And then we found that they're less than or equal to 0.003. Now this has the end user of the cell label peptide will need to assess their own LLOQ on their own instrumentation, as instrumentation variability can certainly play a factor into the LLOQ that's being measured. And then we also measure the linearity of each of these cell label peptides. And then we have our square value of 1.0. And lastly, we used the cell label peptides to quantitate actual LPL and clustering from multiple in-process samples. So all the in-process samples come from CHO IgG producing cell line. When we started with CHO HCF, and then the second intermediate process sample being protein A, and last being IEX. This is a pretty typical clearance steps that's pretty typically seen in CHO, or basically CHO IgG producing manufacturing process, where you see the greatest clearance in right after protein A. And this trend also aligns with our results that we have previously seen in ELISA and DIA data as well. And now we're going to talk about the upcoming CHO no-cell HCCF reference material. So this is a CHO no-cell HCCF. It's a pooled material from two different CHO cell lines, CHO KSV, CHO K1SV, and CHO K1. And the format of this reference material would be at 250 microliter at 1.3 mg per mil. And it's very well characterized both by 2D silver stain, Western coverage analysis against 3 different commercial ELISA kits, as well as HCP, LC-MS, MS analysis by swath. So some of the potential application for this reference material will be matrix and sample for host cell protein peptides in LC-MS, MS, recovery control for protein A HPLC method, as well as control for Western blotting and host cell protein ELISA coverage. Its projected release is in Q1 2025. So for Western coverage analysis that we done on this material, we did Western coverage analysis using 3 different commercial polyclonal antibodies, serums, and the commercial kits of these anti-CHO coverage data came out to be, for the first kit, about 65%. For the second kit, about 76%. And kit C, we observed a 66% in coverage analysis. Lastly, we also performed a swath, basically LC-MS, MS swath analysis on this reference material. So the method that's used here involves sample prep, which used 7 internal standard proteins, reduced with DTT, alkylated with iodocetamide, and digested with lysoendopeptidase, as well as trypsin. And the mass spectrometry approach was separated peptides on C18 column, followed by swath collection for at 350 to 1106, and then followed by analysis. Data was queried against the CHO proteomic database and quantified using an internal standard of response. So from our findings, we identified over 1,138 CHO host cell proteins, with 991 of them above the LLOQ. And total HCP concentration is measured at 900,093,331 ppm. And on the right hand, we have a top 20 list of identified problematic HCP. Some of them are, it doesn't come as shockers, that we have identified them, such as, glutathione S-transferase, protein disulfide isomerase, heat shock protein HSP90 beta, and also pyruvate kinase, peroxyrin doxin 1. These are very problematic host cell proteins. And if you want to find out more information about them, Biopharm has also a very published and a very well published database that you can cross-reference these problematic host cell protein against. And in summary, so USP, we are supporting the evolving field of whole cell protein identification as well as quantitation by mass spec. With the upcoming 1132.1 residual whole cell protein measurement in biopharmaceutical by mass spectrometry, which is posted to PF 49.3 and expected to be official May 1st, 2025. And then we have a wide range of analytical reference material under development. So as you've seen, we have the PLBL2, which is now available. And now we also have the clusterin and LPL cell labeled peptides that are available. And we're also having additional six target whole cell protein with 3 peptides each that's working in progress. And then we also have the pooled HCC sample matrix that will be made available soon this year. And then we also have educational courses on best practices for residual whole cell protein measurement in biopharmaceuticals. So if you're interested, you can scan this QR code, which will take you to our whole cell protein landing page. And lastly, USP as an organization, we are fueled by our stakeholder engagement. And there are many ways to contribute to our mission, such as working in roundtable, working groups, or donating methods or material to support our standard development, or participating in collaborative testing, as well as a review of chapters, stimuli articles on pharmacopoeia forums. So if you are interested in collaborating or contributing or volunteering with USP, please visit our website, because we are looking for candidates and opportunities to join the expert panel or our expert committee for the new 2025 to 2030 cycle. And lastly, we cannot do what we do without USP expert volunteers. So there's a QR code on the left-hand side if you want to learn more about how you can contribute your expertise and knowledge. And that concludes my presentation. And as an acknowledgement, I want to thank my team, Naomi, Anthony, Sarita, who has contributed a lot to this presentation, and as well as our product manager, Shankar, who's developed a lot of these infographics for this presentation that you have seen, really breaking down a complex and abstract idea such as whole cell protein mass spectrometry analysis into something that's more digestible. So thank you. And if you have any more questions, feel free to ask me now. And also here's my email. You can also connect me through here. So thank you. And I'll give it back to Ivan.
Thank you very much, Derek, for an excellent presentation. It's a lot of information to take in. So I think to the audience, if you have any questions for Derek, please type them now in the chat. I can see already now a lot of questions. So I'll just take some of them here. So Derek, do you see a general trend or preference in the industry to data-independent acquisition or data-dependent acquisition?
I think both are, very viable choices, right? It really depends on, what you're looking for and where your starting point is when it comes to untargeted proteomic discovery. So as of now, I think both approaches, especially as we describe in the chapter as well, both data-independent acquisition and data-dependent acquisition are viable choices for the data analysis portion, as described by the USP chapter 1132.1.
Another question. Do I understand it correctly that sample preparation to enrich HCPs in the sample is required before LC-MS? How would that affect recovery or subfractionation of the HCPs? It was one of the approaches you mentioned in the chapter, the enrichment prior to LC-MS.
Yes, so the enrichment process, let me just do a quick check. Wait, could you repeat that question again?
The question is alluding to the native digest, for example, to enrich HCPs and remove the drug substance. Is that necessary or required before you do the LC-MS?
Well, that's actually a good question as well. It really depends on your drug substance and how highly concentrated your drug substance is, right? The more concentrated, your drug substance is, the more that you have the signals, from your host cell protein might be completely masked by your drug substance. So that's really case by case dependent, and it's really based on the type, also based on the type of therapy that you're working with, So that's really up to the biotherapy manufacturer. So if people are working with low concentration, drug substance, that might not be necessary. But, if you're working with something that's very high, it's like 100 mg per mil, the more recent, or like, the high producer cell lines, that might be necessary to do, or you might see some of the signals from your host cell protein being masked completely.
Another question. You mentioned that mass spec requires a lot of technical skills. Can you elaborate on why that is and what skills are needed to use the method?
Well, it really depends on, whether you're doing LC-MRM, or are you doing targeted, or are you doing the untargeted approach, right? If you're doing the proteomic discovery approach, that often requires the knowledge of not just setting up the mass spec part. That also requires knowledge from a proteomic discovery standpoint. You also have to understand a certain point about bioinformatics as well, right? Because whatever you're putting into your most upstream, basically, of the proteomic discovery part, if you don't identify a correct proteomic database, you might miss some of the proteins if you want to later develop for a LC-MRM more targeted approach, right? So it's technical in essence that a lot of smaller, biotherapy manufacturers, or especially startups, might not have the resources or the capability to hire a team of mass spec scientists versus, much larger organizations such as, Pfizer's or Merck. They probably have their dedicated departments, who are basically a department of mass spec specialists or scientists and PhDs. So that's why it becomes challenging, right? Whereas using ELISA, right, you can just buy off-the-shelf a kit. It's very easy to train an analyst to run that. Whereas, the amount of resources that you need to train somebody to do mass spec is quite different, right? So that's why mass spec is typically outsourced as a testing component.
Then we have a group of questions on the standards that you are going to provide. Is there any USP reference protein for E. coli?
Yes, so this is a part that I forgot to mention. So as our general strategy for our host cell protein development, we're currently developing a lot of these for CHO cell lines, but we are dedicated to providing a total solution package for legacy products that are made in E. coli cell lines, as well as the new and upcoming novel therapies. So we're going into E. coli cell lines. We have, plans to go into HEC-293, SF9, Vero for these more novel therapy, as well as more legacy products. That could be something that, like what we have done with the CHO HCCF, or the things that the reference, analytical reference material that we're doing for the cell-level peptides. So we are committed to helping biomanufacturers, who are working with different types of cell lines, and we're committed to helping them through every step of their CMC and development process.
So what I hear, Derek, is that the USP is going to provide more and more standards for
in the future. Yes.
Yes.
For both novel and legacy products.
So another related question: how do you pick internal protein standards if wanting to establish an LC-MS swath method for HCP analysis? I guess that's the intact protein.
Yes, that would be the intact protein approach. So that's a little bit more difficult to to answer. So, well, there's no straight or forward answer for that one, because you have to understand, like when you choose intact protein method or the quantitation strategy, you are quantitating your host cell protein against one, single intact protein, or that intact protein strategy actually has been published, as such as the Eli Lilly method, which uses 3 different types of chicken proteins. So you really have to be careful about like what type of protein that you select and how the protein is made. So let's say if you select certain proteins and you find contaminants inside it, and you're using contaminants being such as E. coli, host cell protein, or E. coli proteins, that could add to, and you are an E. coli, cell line producer, that could add just more to your background signal for your host cell protein that's being measured, right? So a lot of this aspect when it comes to intact protein is really finding a highly pure intact protein that's not part of the recombinant therapy process. This is actually the first time, in recombinant therapy, we're actually trying to find a more naturally sourced protein that's highly pure. So I think that's the difficulty part about using the intact protein part, is finding a naturally pure intact protein that gives a good response curve.
Mm-hmm. Then we have some questions on the potential impact on this chapter and the new standard. So I think the first question is, clearly it's the chapter is written by a group of really experts in this field, and so what's the FDA's liaison? What's their role in writing the chapter?
They're just observers on the panel. For all our expert panel, we always have government liaisons. They observe the meetings, but they do not, write the chapters. The chapters are written by our experts, and neither is USP writing these chapters. So these are written by, experts who are using these methods, these methodologies, and this is like purely what they have come to agree upon as the best practices and moving forward for host cell protein mass spectrometry.
So I mean, clearly the chapter number is above 1,000, so it's not by law that you have to follow it. So, but what do you expect the impact will be on the industry? How much will the best practice be followed by the industry? What have you seen for other chapters?
Yeah, I think this gives like a really good starting point, for the industry, especially in this field of host cell protein mass spectrometry, where there's a lot of clouds over it. It's not well understood. It's a lot of times it's the information, you have to go through many literature searches and read many papers to really understand, what this industry look like, But the chapter provides, basically as an introduction and actually not an introduction, but an in-depth dive for both new and experienced users to really help guide them to select, what is the best practices and what is the best approach for their mass spec host cell protein analysis development. So that's why in the chapters, the experts really left it, to as many possibilities as possible, especially in the sample preparation parts, instead of like focusing on one or combining into one best, but there's not one best choices, right? There's multiple choices that can be used. And it really depends on your fit for purpose, what your need in your lab is, like what your capability are. And then relative and the quantitation strategies that we have described in the chapter as well is, there's three different choices to choose from. It's not there's just one strategies, but this gives at least somewhat of a coalescence around the industries. And as we progress more and as technology gets better, we'll see this idea being coalescent even more, actually.
So when you look 1, 2, 3 years ahead from now, do you expect that FDA will start require that people read the chapter and use the recommended method, or?
That part I can't really comment on, but as from if you dealt with the agencies in the past, there's a common joke is that the agency rather be burned at the stake rather than answer any of the questions. So I don't think the agency will say anything about requirements or requiring, but if you are a CMC specialist, CMC expert, it's about more right? The more that you know about what's in your process, more about the consistency of your process, that gives you more from a risk assessment side, right? It lessens the risk at later stages where you find out there's a host cell protein that might be impacting either your clinical study, or it might be impacting your process scaling or the stability of your product once you go into phase 3 and put on your where you make the reference standard law and you put the reference standard law on stability, and you realize, oh, there's a host cell protein that might be degrading my polysorbate 20 or polysorbate 80 over time. So that's where I see it being more useful. Yeah.
So this chapter is written by the experts and published by the US Pharmacopoeia. So it's a US based, you can say. So how much will this affect Europe, for example, or other countries outside the US? Do you provide the standards outside the US?
Yes, we provide these standards globally. So in the past 10 to 20 years, USP, we are working towards not just providing solutions for our US-based customers. We're also becoming more of a global entity. We are providing solutions for people, for customers where in certain countries or developing countries where there's lack of standards and access to quality medicines. So yes, that's one of our primary mission, is to ensure the safety and access to these quality standards, which ensures the quality and then the efficacy of the medicines.
Mm-hmm. I think we will not have time for more questions, but I can see a lot more questions on the chat. So we will follow up after the webinar. So we have to end it now. But we will follow up after the webinar and contact you directly, and also let Derek answer some of the questions. So with that, I would like to thank you very much, Derek, for an excellent presentation and discussion, and also the audience for very good questions and for your interest in this topic. So thank you very much, all of you.
And thank you, Ivan, for the invitation to give this talk.
Bye-bye.
All right, stay connected.